Hi guys, well, exam is coming up, so these videos are here to help you prepare for your exams. I will start from lecture 1, and these are biology 4, and these notes are for Richard's class. So if you have other teachers, I can't guarantee I can cover everything, but I'll make a separate video to help you study biology in general. For example, how to take notes in class and how, if you're absent, how do you study from just taking notes from the PowerPoint? Okay. Lecture 1 is a review of immune system. Please don't skip this because this is the foundation to prepare you for other lectures. And so what happens in acute inflammation? There will be heat, redness, swelling, pain. In acute inflammation, an example is having a cut on your hand. So when a cut is formed, there are two things that's happening. One is there's clot formation. The system is trying to close up the bleeding. The other one is the system is trying to open up the vessels inside of the cut in order to send nutrients. So more blood and more white blood cell can come to the site and fight for infection. So why there's heat? Because of the blood. Blood is warm, so there will be heat. Redness it's because of the blood. Swelling is because at the site we have white blood cell, a resident white blood cell that's called histamine. That's called mast cell. Mast cell will release histamine, and histamine is a chemical that will vasodilation. Why we need vasodilation is to keep the traffic line open so that more blood, more white blood cell can come. So swelling. Here I wrote histamine that will increase vascular permeability. That's just saying the vessel will be open up. Pain. Why there's pain? Because when we cut, we stimulate the nociceptor. Nociceptor is the pain receptor. All right, that's it for these 4 concepts. Moving on. PAMP and epitope. What are these 2 things? PAMP is characteristics of bacteria. Anything that bacteria has, human cells doesn't, is a PAMP. So like flagellum or cell walls. Epitope is like antigen, the key on the membrane of a pathogen. Moving on. So if you don't understand, maybe search up on Google, and if you still doesn't understand, text me, or you can ask your question under the comment. Better to text me so I can answer you right away. And moving on to this concept. We have APCs. It's a type of white blood cell that will engulf the pathogen and then present a part of their protein on their receptor. So we have three APCs: dendritic cells, macrophages, and B lymphocytes. Think of these 3 as secretaries. What's the job of secretaries? It's to categorize their client or their documents. So we have 2 types of that client. One is from inside, like a cancerous cell, MHC1. Another type is a pathogen coming from outside, like bacteria. So if a bacteria coming from outside that will engulfed by APC, and then they will present part of their protein on their MHC2, and then present it to B cell to maturize the B cell so that it can later produce antibodies. And among these three APCs, the chief of these three secretory is the dendritic cell. Dendritic cell can both recognize something pathogen from the inside, such as cancerous cell. It can also recognize something from the outside, like bacteria. So only this dendritic cell is able to present these two receptors. Okay, that makes the dendritic cell most powerful. I hope you understand. If not, please understand this concept before moving on. Okay. And what I wrote in the light purple is the location of these APCs. This is the whole picture of what happens. When a pathogen comes in, it will engulfed by one of the APC. So let's stay on the upper line here. So a pathogen comes from outside. It can either be engulfed by dendritic cell or macrophages or B cell. And then it will present partial of this pathogen's protein on their MHC2 receptor and present it to T cell. And T cell will be maturized into T helper cell. And with T helper cell, it can maturize B cell into memory cell and plasma cell. And plasma cell will later create antibodies. This upper line is called humoral immune response. It's just a name. Don't overthink, please. And the lower line is when this pathogen, such as a cancer cell, something goes wrong inside of our body and we need immune system to clean it up. So cancer cell will be engulfed by which APC do you think? So only the dendritic cell, because only dendritic cell is the chief who has the ability to recognize something from the inside, MHC1. And with MHC1, it will double link with the T cell and then maturize T cell into T helper cell. Never mind the double link. If you don't understand, I will explain to details later. For now, just know the general process, pathogen engulfed by APC and present to activate T helper cell, and then T helper cell either maturize the B cell if the pathogen is coming from outside, and if the pathogen is from inside of the body, then T helper cell will maturize CD8 T cell. CD8 T cell will later become memory cell and cytotoxic T cell. The lower line is called cell-mediated immune response. Okay. Antibody. These are different functions of antibody because you need to understand what is antibody for. So first, antibody is used to tag the pathogen. Tag anything that's not us. So it's called opsonization. And after tagging something, one cascade will be triggered. That's called classical complement activation. At the end, so it's the pathogen will be tagged by this antibody, and then this Fc portion of the antibody will recruit a complement protein. And this protein will signal other protein, and the other protein, it's like a domino effect. And at the end of this domino effect, the protein will create pores in the membrane of the pathogen, like poking holes on the membrane of the pathogen, and therefore the pathogen dies. So the process of, at the end of this cascade, the protein creates pore in the membrane of the protein is called MAC, MAC. Okay, that's it for the second function of antibody. The third function is called ADCC. Here, for example, we have cancer cell, and the antibody will tag this cancer cell, and it will attract the natural killer cell. And natural killer cell has the receptor to connect to the antibody on the cancer cell, and they will recognize each other and shake hand, grab each other's hand, and then natural killer cell will produce a chemical that's called perforin and granzyme, and it will create pore on the membrane of this cancer cell, and therefore this cancer cell lyses. Lysis means broken, and cancer cell is dead. Next function of antibody is neutralization. If there's toxin floating around, the bacteria continuously secrete toxins. We want to neutralize the toxin, so the antibody will go and then neutralize, prevent this toxin to harm our system. Okay, next function, agglutination of microorganism. If we have multiple bacteria, it's hard for the white blood cell just clean up one by one, so we group them. We group them by using antibody. The antibody is like a glue, and they group the pathogens. And then this group of pathogens will flow through lymph or liver, and then eventually clean out from our system through kidney. Next is precipitation of soluble antigen. So when the pathogen lyses, the cell lyses, there's broken pieces of those pathogen. It's still considered not ourself, so we need to clean it up. It's like the function of glue. The antibody again group these soluble antigens, and then this group will flow out of our system through kidney. Goes to liver first and then clean out from our system. Okay, that's it for the function of antibodies. Do not try to memorize this, it's not important. Just get the idea of, okay, this antibody has these functions. And here I drew a picture of what happens in acute inflammation if you have a cut. So here you have a cut, and then one side of it, the platelets and plasma protein will be present. Platelets will try to form clots and close up this cut. On the other hand, under this cut, we have resident white blood cells, that white blood cells that already present in the local area. And we have three different local white blood cell: mast cell, resident dendritic cell, and tissue resident macrophages. The biologists just give them these three one name to, they just like to give stuff names. So it's called sentinel cells. Not that important for the exam. So you just need to know at the local site, we have mast cell, which secretes histamine. Histamine is used to open up the traffic line, vasodilation. Heparin again, they want to keep the road open. So heparin will prevent clotting. And then chemokines. Chemo means, I'm not sure, never mind. Chemokine is to attract neutrophils, more white blood cells. They need help, more white blood cell to come and help. And resident dendritic cell will kill um, the pathogen if there is pathogen. And also you are able, if I hope you can remember that dendritic cell is the chief of those three secretaries. So it has both MHC1 and MHC2. If you can't understand what I said, please go back to the one before I mentioned, like this section. We also have tissue-resident macrophages. Macrophages is also used to clean up the pathogen. Phagocytosis, eating the pathogen. And it also releases cytokines. It's toxic chemicals. Let me double check. Cytokine. Oh, cytokine is to attract more white blood cells. And then what's coming later is the neutrophil. Neutrophil will later come, and on top of that we have monocytes. Monocytes is just another type of white blood cell that will later becomes macrophages. It's like an upgrade version. These macrophages, so why it's given another name, it's because we already have macrophages at the local site. In order to differentiate these two, they give the later coming, the one that comes later, another name. It's called monocyte-derived macrophages. And in the meanwhile, everything of this is happening, we have the complement pathway. It's the cascade I mentioned before, because there will be antibodies at the local site, and that will activate the complement cascade. So here's the order you can read on by yourself. I'm moving on to lymphocytes, the story of B cell and T cell. So both B and T cells are born in the long bone marrow, long bone, and it's in the red bone marrow. And after it's born, B cell will stay here, and then T cell moves to thymus, and they are going to be trained. So what are they training for? They're training for, first, they won't grab the hand of their friends, which is called positive selection. And secondly, they need to shake hand of their enemy. So you see the key connects means I'll just refer to grabbing their hand. They want to grab the hand of an enemy and do not want to grab the hand of a friend. Negative selection. After education is done, they move to work. So where do they work? They work in the lymph node. Once they arrive in the lymph node, they will be maturized by the APC that presents the protein, a piece of protein of a pathogen on their MHC1 or MHC2 receptor. Now I'm moving on to the process of how B and T cell is maturated. So let's talk about B cell first, the humoral immune response. So APC presents MHC2, means the pathogen is from outside. And then with MHC2, it will connect with CD4 T receptor. How do you remember this? It's how I remembered it. It's because back then my friend was 18 and I was 24. So 2 goes with 4. They connect. 2 always goes with 4. MHC2 connects with CD4, and at the same time, APC release the cytokines to activate this T cell into T helper cell. And T helper cell will also, one of its receptor will connect with B cell, and then T helper cell release interleukin-2. So in the exam, the teacher doesn't want you to remember which interleukin, just say cytokine. It releases cytokine to activate B cell. So 2 things has to happen. 1, the receptor connects. 2, the cytokine is released. And B cell is matured into memory cell and plasma cell. And plasma cell will later create more antibodies. So in the exam, I recommend you to draw this picture, or you can write this equation. APC with MHC2 plus CD4 T cell. This CD4 T cell will become T helper cell. And with T helper cell connects with B cell equals plasma cell and memory cells. Okay. So we are moving on to if there is pathogen from our inside of our body, like a cancerous cell. So APC will engulf the cancerous cell and then present a piece of its protein on their MHC1. And MHC1 will connect with the CD8 receptor on a T cell. That is like I mentioned my friend was 18 back then, so 1 always goes with 8. 18. 1 goes with 8. On top of the connection with this 2 receptor, we have the APC releasing cytokines. And then on top of that, we also need the T helper cell to release this interleukin-2, this cytokine. So we need, like, when you think of it, it's like, okay, we're in the process of activating the CD8 T cell, but we also need on the side, when T helper cell is triggering the activation of B cell, the cytokine it releases helps with the activation of CD8 T cell. So we need 3 factors to activate the CD8 T cell. 1 is MHC1 has to connect with CD8 receptor. 2, the APC with MHC1 has to release the cytokine. This cytokine is different from the other one. This one is interleukin-1. And third, we need this cytokine that's released when helper T cell is activating B cell. So needs 3 factors to get maturation of CD8 receptor. After that, this T cell will become cytotoxic T cell and also memory cell. So this cytotoxic T cell is pretty toxic. It's in its name. It creates perforin and granzyme that creates pore in membrane of the cancerous cell, something from in our own body, and the cell will lyse. will perform lysis. So the equation will be APC MHC1 plus CD8 T cell and plus 2 types of cytokines times 2. 2 different cytokines to have cytotoxic T cell. Now we're moving on to this one is a repetition of what happens. Oh, okay. Yeah. Here I wanted to mention something that's tricky. So you know that B cell is one of the APC, the 3 secretaries, B cell, dendritic cell, macrophages. So B cell is one of the APC. So if a B cell engulf it so it connects with the epitope of a pathogen that's from outside, and then this B cell will connect with T helper cell. The activation of T2 goes with 4, so CD4 T cell becomes T helper cell. And then this T helper cell is able to activate the same APC that's also B cell. And this B cell can become memory cell and plasma cell. So it's something tricky about the B cell. Now I'm moving on to antibody class switching. So it's not very important for you to memorize. Just go through it so you know that which one is which. You just need to have a general idea. Okay, antibody has different classes, and that's it. Not too complicated. No, you don't need to know which type is for which. When it comes up later in the lecture, you will get to know them. And for the bacteria, we have gram-positive, that's purple, has multiple layers of peptidoglycan, and the gram-negative, that is pink, that looks like this. Okay, moving on to lecture 2. So lecture 2 is about hypersensitivity type 1 and hypersensitivity type 2. Type 1 is the allergy. So for the first time, I'll start at the beginning of the story. For the pollen, for example, is coming into the body for the first time, and it will be recognized by APC of its PAMP. PAMP is the characteristic of a pathogen that has something different than our human cell. For example, cell walls, flagellum, and or peptidoglycan. And APC engulf this pollen, for example, and then it presents it on the MHC2, and MHC2 will activate B cell. Then we have plasma cell, memory cell, and plasma cell will create antibody. So now the antibody, the FC portion is flowing. So this is happening in the lymph node. And then this antibody will flow to the surface of the skin or to the local site. And then this FC portion will stick to the mast cell. So for the next time when the pollen comes in, the allergen comes in, the Fab portion will able to recognize the allergen really fast. And then because it's connected to mast cell will start secreting histamine. Why is that? Because they are recognizing the pollen as an enemy. They're they're sticking to it to tag it. And the purpose of tagging it is to recruit more white blood cell. And if we recruit more white blood cell, we need to open up the highway. So mast cell is secreting histamine. Histamine is to increase the permeability of the vessel. And why I put basophil is when we have a large quantity of allergen coming in, and after mast cell is triggered, eventually basal cell is also triggered. When basal cell is triggered, it's like an anaphylactic shock. And what does allergen cross-linking mean? It means when a small amount of allergen comes in, when it only sticks to one side of the fab, it doesn't trigger a response. It has to be big enough and multiple fab is connected to this allergen, then the mast cell will react. That's called allergen cross-linking. And different response in different effector is I put it here is trying to say that if this mast cell is releasing histamine at the trach, it will cause bronchoconstriction. If it happens under our skin, then it will be vasodilation. There will be swelling. So moving on to type 2. Type 2, a key difference is that there is already antibody. And the antibody, the Fab portion, right now is not the FC portion. It's the Fab portion that stick to the pathogen first. And then it recruits natural killer cell and performs ADCC. If you don't know what ADCC means, please go back to earlier. And besides that, that's number one I marked here. That's one thing that could happen. A second thing that could happen is because this antibody is sticking, it's tagging here, it will cause the activation of classical complement. And classical complement will lead to more white blood cell coming, or it can lead to MAC. Now I'm going to explain one example, the first example of type 2 hypersensitivity. Why blood transfusion is type 2 is because our body already know which type of blood we have, so it pre-make antibody. There's already antibody in our body against other type of blood. So if we have A, we will have antibody B. And if we have B, we'll have antibody A. I think everyone gets it now. It's at the end of the semester. And let's talk about RH. So if this person is A negative, so there's no RH on this, naturally they want to go against RH. It counts as an enemy. So the person, if it's for the first time the person receive RH, they will develop anti-RH antibody. So before the first time, they actually don't have RH antibody. They have to get contact with this RH blood first and then develop anti-RH antibody. And if the second time they receive any blood, even A type blood, but with RH in it, these antibody will be triggered. So in the exam, always I recommend to draw the person's type of blood and draw the antigens on the blood and figure out which antibody they have first before trying to answer the question. So these are different types. And for AB, they don't have any antibody, just the RH if they receive RH blood before. O, they have both A and B RH here if they received RH before. And now I'll talk about ROGAM. ROGAM is given to moms who has RH negative. So the logic here for ROGAM is at the end of a delivery, if this mom is giving RH plus baby, giving birth, and then at the delivery, when she's delivering this baby, some fetus blood will remain behind. And that will cause this mom to develop our anti-RH antibody. And if this mom get pregnant again with a RH positive baby for the second time, then that baby will be killed because the antibody, our anti-RH antibody is small enough to transport, moving. They will able to be moved to move inside of the placenta and kill the baby. Baby will die from anemia. That's why at the end of the first delivery, we give ROGAM. ROGAM is like an artificial antibody. They will perform neutralization. They will also perform agglutination and then clean this fetus blood out of the mom system. Okay. Any questions, you can text me if you don't understand this part. So anti-RH antibody is small enough to move into the placenta, and the other blood antibody is too big. It's IgM, so it won't be able to move into the placenta. So it's not a concern that the baby has different blood type, but it will be a concern if the baby has RH positive and the mom has RH negative. Second example of type 2 is when there's hapten and hapten bind with a protein inside of our body that makes the antibody recognize it as a pathogen. You will later see hypersensitivity type 4 also involve hapten. The key here, which makes this thing type 2 reaction, is because there is antibody. This antibody comes from B cells. The maturation of B cell into plasma cell, and plasma cell produce IgG. Produce antibody, and this antibody will the fat portion connects with the pathogen, makes it type 2 hypersensitivity. Third example, non-cytotoxic reaction. First illness is myasthenia gravis. Don't be scared by the name. It's talking about the weakness of muscle. So on our skeletal muscle, we have acetylcholine. This receptor in normal time receive acetylcholine as a signal to contract. But here we have antibody that blocks this receptor and prevents it from contracting. So that's why that leads to muscle weakness. And Graves' disease is when antibody is connecting to the receptor of thymus cells on the thymus. It connects with the thymus and stimulates it to produce more thyroid. So at the end of the day, Graves' disease leads to high thyroid hormone. Cross-reactivity means If a antibody develops with by the ability to shake hand with an enemy, however, this key is so similar that's going to shake hand with our friend. So that's autoimmune disease. That will lead to autoimmune disease. So this is called cross-reactivity. And here's an example of question. So if the patient has A negative and this patient has never received Rh blood before, what type of antibody does this patient have? So the answer will be antibody B. There won't be any anti-Rh antibody because this patient, I said, has never received Rh blood. I think that's it. Oh, okay. Also, for the blood type, if we are testing a person which blood type they have in the lab, whichever that turns color or react to what the laboratorist drop into the plate is the type the person has. Because they're dropping an anti-A or anti-B. So whatever they drop in is and the blood react to it is the type the person has. Okay, lecture 3. Type 3 hypersensitivity. So before explaining this, I will tell you what happens in normal healthy person. And in healthy person, when a pathogen lysis, the small pieces of that pathogen will remain in the system. So our system need to clean these trash up by the antibody agglutination and then form a group of these and soluble antigen, and then they flush to the liver, and then kidney will excrete it or through lymph nodes. But however, in type 3 hypersensitivity, this happens is because the person's immune system is not alert enough. They don't clean up fast enough so that these antigens remain in the body. And then, so I'll start from the beginning, this process, antigen remain in the body, and then APC comes in and engulf it, present with MHC2 receptor, Y2, because B cell is activated. B cell only is being activated when the pathogen is coming from outside. T helper cell activation and activate B cell, we get plasma cell, memory cell, antibody. Antibody goes perform precipitation, and then because this person's immune system is down, it's not completely down, it's still working, but it's not fast enough. So this precipitate group flows around and end up in capillaries, anywhere that has small vessels like joints. And then they stick to the tissue of that joint, the capillary. And then because it stick to it, it's still a pathogen though, this group of precipitation that will attract neutrophil. And neutrophil will release chemicals, toxic chemicals. They have good intention. They are trying to get rid of this pathogen group. However, because it's sticking to the tissue and the chemical just spreads and it will damage the tissue nearby and cause inflammation. In the meanwhile, more neutrophil will be accumulated and more because there is damage to this tissue. So mast cell will also release histamine to keep the traffic road open. There will be that vasodilation and more neutrophil comes. This is a vicious cycle. And then neutrophil continue secreting these chemicals. Then the tissue is damaged. Because when tissue is damaged, we need to fix it. The body just naturally secrete collagen fibers and platelets. And because it's in small vessels, capillaries, and these fibers, collagen fibers and platelets, clots will decrease the blood flow to the joint. And then the tissue becomes necrotic. All right, type 4 hypersensitivity. The story goes, you walk in a forest and then the poison ivy gets into your body. In lucky times, the hapten means just a piece of that protein, ivy poison, for example, just flash out of your system. Nothing happens. Unfortunately, if this hapten bound with some integral membrane of the protein in your body, then they form a pathogen. Part of this pathogen is yourself. Part of it is from the poison ivy, so it's something from outside. So both of these receptors will be triggered, will be presented on the APC. APC that has MHC2 is three of the secretaries, and APC that has MHC1 is only the dendritic cell. And in type 4, focus more on this CD8 T cell, this line. It's the cytotoxic immune response. You can go back to earlier to check out the name. And the CD8 T cell is activated, and that will become cytotoxic T cell and memory cell. And because this whole process takes long time, so the person won't react to this hapten pathogen too fast. So the cytotoxic T cell, when it's developed, it's going to cause some damage. Because remember, the protein is from our cells. So the antibody that's going to create by this memory cell, well, they will cause cross activity. So it's not only able to recognize this pathogen, it can also shake hand, grab the hand of our own cell. Let me check. Cell-mediated immune reaction. So the key point here is that in type 4, it's the active maturation of the CD8 T cell. It's the cell-mediated immune response. All right, that's it. Good luck. I will keep up with the videos and finish soon. Ask me questions if you need. Bye.